So our next topic is related to chromosomal aberrations and problems related to chromosomal structure. So far we have seen how important it is, how chromosomes they are arranged and they are processed for either replications or the what we call it as a gene expression, that is RNA protein synthesis. Now next is we have seen one more important thing is everything is very important. There are so many factors are there. There are transcription factors are there. They are responsible for processing gene or expression of gene at certain time, that is less or more time or less or more in quantity. So the timing, schedule, amount of protein, everything will be decided by whole this mechanism, and on that basis these proteins, either structural or functional proteins, these will be responsible for various anatomical or physiological roles the cells are playing. Now anything goes wrong with that, if the system is so good, we have seen that even we have got gene repair mechanism, if you remember, during replication. Okay, and there are various checkpoints are there, and during checkpoints there is a identification of a problem, and then this problem is rectified. So such a good system we have. Still we find diseases in people, so many diseases are there. So why this? So let us see various reasons. We'll start with what are these and how the things are there. So let us see that why this happens. This is happening because of changes in the genetic material. Now why genetic material should change? So it may be because of any internal or external activity, it will be there. Okay. So whatsoever changes are there, and these are permanent, we call it as a mutation. These are the permanent transmittable changes. That is, they are transmitted from one generation to other generation. So these are the happening, and usually these mutations are happening in the single genes. Now that means that the genes which are parent genes and the daughter genes, they will have differences, because now the daughter genes has got mutated. Now the next thing is that we can find so many structural differences. If you see the people, we have got Asians, we have got Caucasians, we have got Americans or Eurasians or Europeans, and we have got African people. We can see drastic changes in their overall appearances. Okay. Now why this is? Because we call them as a genetic makeup which is different. So this is because of the changes, and these changes are known, and for that we call that these are the polymorphisms. Polymorphism means the genes are having different kind of genetic setup, and because of this polymorphism, that what you will encounter in 5th year, pharmacogenetics is a branch where because of the polymorphism there are people who are having either lesser or more active gene. If you we shall go for this topics of chemotherapy, in that we shall see some of the drugs are there, some of the chemotherapeutic agents, they are rapidly metabolized, some are less metabolized. So why this happens? Because again this is a polymorphism. Because of this polymorphism, what will happen is this polymorphism will cause example, in case of if you remember paracetamol, we call it as a slow acetylator, fast acetylators. Similarly, it happens in isoniazid, one of the anti-tubercular drug. So these are the acetylations are there. Acetylation is the normal metabolism process, drug metabolism process in the body. But because of the polymorphism, in some people it will be fast, in some people it will be slow. And accordingly, the outcomes will be there. If it is fast, then drug will be metabolized faster. the action will be wearing off faster. In slow, it will be remaining for longer time. Depending on the metabolite, if the metabolite is toxic, then fast metabolite, these fast acetylators will have problem, or if the drug is toxic or drug is having adverse effect, then slow metabolizers will have a problem. And normally if the drugs are drug dosing are calculated on only one kind of time, then the other will face any kind of problem, either a rapid this deactivation and therefore rapid wearing off of the effect, and otherwise it will be causing the accumulation of the drug. So these are the problem. This is what we call as a polymorphism. Now next in that is what are various types of mutation. We have seen what mutation is. Mutation is there is a changes in the genetic sequence, and basically or usually it is only one kind of gene. So these are variety of this time. Example of these are the genomic type, where the chromosome number will change. Example of these are called as a aneuploidy. Aneuploidy means the chromosome number will change. Example, we know that there are 46 chromosome or 23 pairs. Okay, now these pairs are there, 23 pairs are there, 1 to 22 and 23 number will be 6 chromosome, X or Y or XY, and accordingly this aneuploidy is there. They may have either instead of 46 they may have 45 number of chromosome or 47 number of chromosomes. Then monosomy means instead of a pair, we have seen that up to 22 there is a pair. So this pair is there. Instead of pair, there is only one chromosome is there. We want that chromosome number 1, chromosome number 7, it should have pair of it, that there should be duplication or two things. Here instead of that only one set will be there, that will call as a monosomy. Then polysomy means instead of 2 there will be more than 2. It can be trisomy, that is 3 sets of that particular number of chromosome, maybe chromosome number 3, chromosome number 4, chromosome number 5, whatsoever it may be. So this is called as a trisomy. If it is more than 3, example if it is 4, then we'll call it as a tetrasomy, or any such increase in the number we'll call it as a polysomy. Then another kind of mutations they occur in the chromosomal, that is these are because of the abnormal chromosomal structure. And lastly it will occur in the gene structure, that is changes in the DNA sequences in the specific gene. Even it can occur in the RNA, we call them as splice variants. So these are at various places, these mutations are there, and because of this mutation the outcome is there. We know, we call it as a genotype. So these changes in the genotype will cause changes in the phenotype. Phenotype means functional changes. Genotype means changes in the DNA or genetic material, and phenotype means changes in the outcome, structural or functional. So because of these genotypic changes, there shall be changes in the phenotypes. Now we know how chromosomes are there. We know that these all DNAs are there. They are wrapped around histone, and we call this one set as a nucleosome. And there are variety of these proteins are there, H2A, H2B, H3, H4. These are there, and these are they are forming a reel-like structure around which the core DNA is wrapped, and there is a tail-like structure that is we call as a linker DNA. Now when they are compressed, then we have the chromosome, and this chromosome have 3 parts: telomere, centromere, these are 2, and the center is there. Let us see this. So this is telomere, upper one, centromere is there. Now if it is the centromere is exactly in the center and both arms are equal, we call it as a metacentric. If there is a one arm is a short and another arm is a long, example short is called as a P arm and long arm is called as a Q arm, then we call it as a submetacentric because it is not exactly half division. And lastly, if very small portion is there, and long big tail like is there, or big arm is there, and the another arm is very small, almost negligible, then we'll call it as a acrocentric. So these are various nomenclatures it is having. On that basis, the chromosomal location is defined, because somebody has to tell, somebody has to mention, suppose, or maybe in the genomics people, they will tell that why this kind of disease is occurring. So when this location has to be given, this location is given in a very typical way. So we have seen that there are two arms, P and Q arm. Then there are regions are there on this arm. Okay. So these regions are placed on this arm. Then there are bands on this arm. So in that particular region, one band will be there, and there will be some small sub band will be there. So this chromosome, suppose it was a chromosome number 17 out of this 22 number, 23rd is a sex chromosome. So out of that, this 70 number chromosome, so how people will tell it? That is 17 Q 1 means region number 1. See, chromosome number 17, it is Q, that is arm is Q, either P arm or Q arm. Then region number is 1, sub region or band number is 1, and sub band is 2. So then we classify it is in this way. Accordingly, this will be the nomenclature. This is used to inform that what kind of genetic changes have occurred at what place. Whenever the scientists they are trying, they find something unusual, and they linked it with the engine. At what place the problem is there, that is found out. Then next in that is the chromosome morphology change. We have seen that chromosome morphology changes during cell division. DNA double helix structure is there. It is around 2 nanometer diameter. During interphase, particularly in G1s and G2 phase, it is around that. Then we know chromatin is having a bits on a string structure, that is around 11 nanometer nucleosome type structure. Chromosome in a nucleosome, it makes around 13 nanometer, particularly during metaphase. And extended metaphase, it is having 300 nanometer, and condensed metaphase chromosome will be around 700 nanometer. These structural changes, they are occurring throughout the cell cycle, because we have seen that it has to be released for replication, and accordingly when it is released, then it is spread, it is increased, and then at the end, when everything is over, it is condensed. We have seen cell division cycle. We have seen various phases, G1, S, G2, and M phase, that is mitosis phase, in which prophase, anaphase, metaphase, telophase, these are there. So these are the changes. So during interphase, it is around 11 to 30 nanometer fibers are there, while in metaphase there are 300 to 700 nanometers fibers are there, which are condensed. Now how it is there, how these geneticists do it? They try to stain them, and the patient cells are intubated, and they are divided in the tissue culture. These are the techniques. Just like those who have studied microbiology, they have seen that there is agar plate or something like that. We call it as a media. So similarly, there is a tissue culture thing. In that, there is a media is there. The cells are there, and cells are allowed to multiply. And then various staining techniques and various chemicals are using. Example, phytohemagglutinin is there, which will cause cell division stimulated. Then colsemide is a chemical which will arrest the cell in metaphases. Then 3s2 methanol acetic acid will cause fixing of the metaphase chromosome for staining. So these all various structures are material are used, and accordingly then next thing is there various stains are used. I think just a minute, because of this color I think you'll find problem. Yes. So these are causing a problem. See, these are the Gimsa is a strain. So this Gimsa strain, that is staining, will show you this type of bonding. So because of Gimsa, it is called as a G bands. Then when these bands means just like if you know phase contrast microscopy and such thing, that this portion should not be stained and other portion should be stained, then we call it as a reverse band. Reverse means, see, you can compare with this. These bands earlier were not stained and these bands were stained. Now these bands are not stained and the bands which were not stained with the G bands now are they are not stained, now they are stained. So this is there, and other are there, only centromere is stained, okay, during metaphase. So we call it as a C band, and these are the various bands which allow you to analyze or think about this various thing. Now karyotype is again this is the international nomenclature is there, how to describe it. So international system for human cytogenic nomenclature is there, it is called as ICS, ISCN, that is International System for Cytogenic Nomenclature. There are genes are there, example 46 as I told you, and XX is a normal female, XY is a normal male, where the G bands chromosomes are identified by their band pattern. These are various chromosomes in normal female. Okay, it shows staining, that is G banding Gimsa stains, that is high resolution Gimsa stain. These are there. Okay. Now let us see various chromosomal abnormalities. This is there in our syllabus. That is, see, these are the pairs. Okay, these are various chromosomes. Now translocation, what has happened is, from this color you will be able to understand it, that this is one chromosome, this is another chromosome. Some part of this chromosome has got into another chromosome and this has got replaced. If you remember cell division, then next, that is meiosis is there. In that similar thing is there, you call it as a crossover. But here this is not a crossover, this is actually for chromosome. There it occurs in single one, that is when two haploid cells come together, then it come or then it is resulting into making this. So this is a translocation. Then deletion, you can see that here some of the portion is removed, it is not there, it should have been there but it is not there, that is called as a deletion. Then next is called as a insertion. So this was a chromosome, both were same, but this much portion, some other chromosomal portion has been put into it, that is called as a insertion. Inversion means they were same earlier, but these structures are there, particularly if we say that chromosomes were metacentric. Okay, so in that cases they are, instead of aligning parallelly, they are aligning in a cross way. That is here one portion, that is P portion is upside and Q is downside, like that. So here vice versa. Then isochromosome is there, that means these are instead of conformation changes is there, the conformation is much different than it should have been normally. Then next in that is derivative chromosome, that is a single one, instead of pair it is there, and again it has got some another attachment, and the chromosome is forming a ring instead of tails which are there. Here we have seen it has got a tail, so instead of tail it has formed a ring. Ring chromosome as such, we have seen that they are present in microorganisms or such kind of smaller type of species. So what are the abnormalities? Now if we compare with the previous one, like that, you can see they are correct one, they are almost straight. Okay, no much changes there. Here you can find that there are changes. Okay, example is translocation is there on the chromosome number 45, okay, XY chromosome, and this is the place, that is number 14, chromosome number 14, Q21Q. So these are two molecules. We have seen how it is nomenclature is. So on that there is some portion is added which is normal chromosome, and there is a translocation on that. So these are various kind of chromosomal abnormality. These are actual staining figures. We have seen earlier are the schematic representation, and these are actual stain figure are there. These are various techniques which are used. Actually we shall be seeing other techniques also as and when they comes. So this is called as a fluorescent in situ hybridization. This is in short form called as a FISH. F for fluorescent, then I for in, then situ. In situ means indirectly hybridization. So hybridization of complementary gene or region specific fluorescence probes are there. You know complementary gene, we have seen this during replication, we have seen this during RNA formation, that whatsoever gene is being processed, then complementary gene is formed, and that complementary strand is formed, ATGC, you know, or when RNA is there, we instead of T it is U. So this complementary is there. Here the complementary is artificial, it is not in body. So this has got fluorescent probes are there on this, and when this is come together, so during that on slide or in situ it is there. So microscopically one can examine it, what is happening, because whenever it is there, whenever it is processed, then all these figures, all the images can be there. This technique is widely used. It is used for metaphase FISH, that is fluorescent in situ hybridization. It is for, we call it as a chromosome painting, that is to understand what it is. Then spectral karyotyping, what kind of karyotype it is having, what kind of structural features that chromosome is having. For that it is used. Then for what it is used, again, identification and characterization of numerical and structural chromosome abnormalities, how many number and at what structure the abnormalities are there. Detection of microscopically invisible deletions, if some of the deletions have taken place. We have seen that what deletion means, some of the part of the chromosome is removed, okay, because of some external or internal reasons. Then it is able to detect the subtelomeric aberration, the aberration which are below telomeric. Then this aberration means abnormal changes. So these aberrations can be detected. Then prenatal diagnosis of the common aneuploidies. I can just give the example. One of my postgraduate student around 10-15 years back, when he got married, around more than 10 years back, and after that while discussion he told me that he is going to have a new guest in that, and later he told me that that was a problem, and it was a trisomy case. So trisomy, just now we have discussed what trisomy means. There is a additional instead of pair, there are 3 sets of chromosome, maybe chromosome number 16, 17, 18, that is called as a trisomy. So this kind of techniques are used to that, and when this it is there trisomy, then there are chances that the fetus or the whatsoever the birth takes place, that male or female baby, that will have genetic abnormalities. So fortunately this can be detected, and it can help to plan the future generation accordingly if the problems are bigger one. Then there are various FISH probes are there, that is chromosome specific probes are there, chromosome painting probes are there, and unique DNA sequence probes are there. These are used for various purposes, and this can be used to detect the finer things in the DNA. Then next in that is FISH probes. Will not go into this detail more, because these probes, how they are there, they are hybridizing the subtelomeric region, and they are detecting subtelomeric detection and rearrangements, and accordingly these are at various places, as we have seen, chromosome specific, and at various places they are doing. So these are helping to understand what are there, if any rearrangement in the chromosome is there, any deletion is there, and thus it will be helping to understand the things. So it will show you various chromosomal signal. I was telling you, see, this is normal diploid signal, that means there is a set of chromosome. But if it is trisomy, that is it will show the 3 dots or 3 stains of that, or 3 fish signals we call them. Okay, so this is giving trisomy information. Monosomy or deletion, that is instead of 2 there is only 1, that means 1 chromosome has got deleted. So that shows indication that 1 chromosome is not there. So this is how these signals help to understand the chromosome things. Structural abnormality also it will be detecting with the fusion probe, and this structural abnormality by interface probe is there. So whether any portion has been broken apart, that has to be checked. It will be give example of that, how this normal is there and how abnormal is there. See, in normal you find that it is a continuous one, it is almost close to each other, while in abnormal they are far away from each other. So this is how this technique is used, and to detect. Why I took this is, this will help you to understand how the things are. This fish and all these techniques, that the details are not there, you just remember that these are various techniques and for what purpose it is there, because we are not, as I repeat again, we are not geneticists that we should be having all these details. Now next in that is how many types are there. We have seen earlier it can be chromosomal at level, or it can be genomic, it can be at the DNA level, it can be at RNA level. So at variety of level it is there. We shall studying one by one what kind of these are there. We all call them as a genetic abnormalities. So we shall see various genetic abnormalities into it. I'll add another one. So these are various genetic disorders are there, and why there can be genetic disorders, abnormalities there. There are many reasons, many what we can say reasons are there why this should be there. Now it can happen during embryonic development, or it can occur spontaneously before embryonic development, and it can be inherited from 2 parents who are carriers of faulty genes. It can be autosomal recessive or autosomal dominant. Now what is difference between autosomal recessive and dominant is, recessive means they are having but they don't show any kind of signs, while when one of the parent is showing the signs of the disorder, we call it as a dominant inheritance. Okay, you may be knowing there are many genetic disorders including thalassemias, major, minor, if you have gone through. So if there are people who do not show any sign and still they carry a faulty gene, we call them as a autosomal recessive inheritance, and from that parent which was not showing any sign, while the anyone who is showing the signs of the disorder, we'll call it as a dominant inheritance. Some disorder are caused by mutation on the X chromosome, while some are called as a on Y. On Y actually very less, X are more. Anyway, X is more dominant because 2 sets are present in female and 1 set is compulsorily present in males also, and Y is only 1. And very few disorders are there inherited. They are because of the Y chromosome or because of the DNA, microsomal DNA. There are known 6,000 or more genetic disorders are there, and still new are being described. And again, for many diseases, the genetic linkage is being found out. There are many diseases earlier known, but now it is found out that they are because of some genetic changes. And although we call them as a even atherosclerosis, like they may be because of certain genetic changes the people have got. Around 1 in 50 people, they are affected by a known single gene disorder. 1 in 50 is not a small number. Okay, that means so many people are there. 1 in 50 means almost what you can understand. There is one person in almost in a class of B Farm which has got a 60 student. So one student is affected by a known single gene, while 1 in 263 are affected by a chromosomal disorder. Around 65% people have some kind of health problem because of the congenital genetic mutation due to significant large number of genetic disorder. 1 in 21 students, people are affected by genetic disorder classified as a rare, which is very less in number. Okay, this is it is rare means defined as affecting less than 1 in 2000 people. Most genetic disorders are rare in themselves. Cancers are called because of the genetic mutations, but generally omitting when we call genetic disorders, because they are not hereditary. Usually, if they are hereditary, then we prefer the use of word genetic disorders. Okay, but yes, cancer has got link with the genetic material. So, how many types of various kind of there? See, these are the example is hypercholesteremia. Okay, these are the single gene disorders where these are autosomal dominant disorders are there, polycystic kidney diseases, hereditary spherocytosis, and many such. Huntington disease, they were not earlier. Huntington's was now it has been being clear that this is because of a genetic problem. So these are the single gene disorders. We can call them as a monogenic, and because of the problem of the genetic structures which are passed because of the in subsequent generation in various ways. So these are the various kind of type of problems. Example of these are many. This can be dwarfism, then achondroplasia, that is a problem in the chondrocytes, sickle cell anemia, then malaria is having more resistance to malaria in some of the conditions. It is because of the genetic condition only. Then autosomal dominant are there. I told you recessive and dominant. Dominant means already problem is there. There are many such there. Example of this: Huntington's disease, neurofibroblastomas, Marfan syndromes, then non-polypsosis, colorectal cancer, etc. Autosomal recessive, okay, where the copies are there, and these people are having no sign symptoms, and these are showing sign in the offspring. Means both parents may be having faulty genes, but they are not showing any sign symptoms. But as soon as the next generation is there, then this generation will get this addition of this both, and then the gene will be faulty and will able to show the sign symptoms. So there are many such there. Example of these are albinism. Albinism is a disorder where the people are totally white. No black, almost look like European, although they are Asian or from similar genetic makeup like us, but they are totally white. It is called as albinism. Then acetyl-CoA dehydrogenase deficiency, cystic fibrosis is one of the very prominent disease. Sickle cell anemias and many such diseases are there. Then a very interesting figure is given in this. I would like to explain this. That is, see, these are various chromosome numbers from chromosome number 1 to 22, and then X and Y. So 1 to 22 are in a pair, and X and Y are there. each chromosome is linked, it has been found, and DNA test is available for that particular disease. Suppose it is known that parents were having one or other disease, then the test can be done for that. So these are various kind of diseases which are linked with particular chromosome number. So we can see one number from that Gaucher disease to Ehlers-Danlos disease, then retinitis pigmentosa, making problem with the retina. Huntington disease, Huntington you might have studied along with myasthenia gravis. Then familial polyposis, that is intestinal polyps are there which causes colon cancer. Then sixth number is calling hemochromatosis, abnormal high absorption of iron from the diet. Like that, there are many such diseases are there which are genetic. Sickle cell anemia is linked with chromosome number 11, phenylketonuria, that is in 12, and like that. Alzheimer with gene chromosome number 14, and there are such multiple genes are there, or multiple chromosomes are there which are responsible for one or other kind of genetic disorder. So as I told you, there can be X-linked dominant, X-linked recessive. Dominant, again dominant recessive we have defined. Dominant means where it shows signs, symptoms. Recessive means there is a fault, but that is not big enough to show you the identification. These are the recessive one. Y-linked are very rare. They are because of the mutation on the Y chromosome, and they are only transmitted from heterogenic sex, that is male humans, to offspring of the same sex, because male to male. Because here Y to Y only possible when it is going to father to son. Father to daughter cannot occur because problem was there on the Y chromosome, and now the daughter will have both X chromosome, no Y chromosome. Okay, so that is there. But X link, why they are more? Because again there even daughter or son both can have X, and therefore that chromosomes can have more impact, which are X linked one. Okay, the females can never be affected, as I told you. Y-linked disorders are extremely rare, but well-known example of particular infertility in some of the men, and in that cases then the fertility can only be possible if other techniques are used, like in vitro fertilization, etc. The third type of gene are there. We have seen the chromosome we can have, that is 2 we have seen X types chromosome, Y type, and third important are mitochondrial. We have seen that mitochondria has got separate DNA so that it can have its own machinery synthesized in the DNA only. So these are the inherent, this mitochondrial is there. It is also known as a maternal inheritance. It is the rarest and applies to 13 genes which are encoded for mitochondrial DNA, and only X cells they are contributing to mitochondria to develop in embryo. Only mothers who can pass the mitochondrial DNA condition to other. Recently, during these Navratris, people have put it this on WhatsApp. I don't know, somebody has got it or not. That why the goddesses are called as Adi Shakti or Shakti, like that. So because all the human get their power from mitochondria in form of ATP, and all these human being, whether it is male or female, they get the mitochondrial structure or inheritance from the mothers. So all whatsoever power or ATP somebody is generating, it is because of the inheritance from the mother genetic structure. Okay, so this was kind of WhatsApp message I could read, and this is how people are applying sciences at different places. So these are multifactorial disorders are there. It can be complex, it can be multifactorial, that is more than one factor is responsible for that. It can also call as a polygenic, that is more than one place of genetic structure are there. They are likely associated with the effect of multiple gene in combination with the lifestyle and environmental factor. Therefore we call it as a multiple this lifestyle and multifactorial one. Okay, because there is a small problem in genes, but when it is coming in contact with some other factors like environmental factor or lifestyle, then these genes or the problem which was there or the fault which was there, that fault gets multiplied, and then the signs are there. Otherwise the gene should have been remain recessive, but when it is there, that becomes it shows its this one. Example of these are heart disease, diabetes. I told you just now that heart disease, diabetes, nobody thought that it can be initially few years back, rather few decades back rather, heart disease and diabetes can be they were thought as a metabolic or some functional disorders, but now we have just seen that hypercholesteremia is a genetic disorder, diabetes, and there are many such things are there which are there. There are many diseases are there like asthma, autoimmune diseases, cancers, then cleft palate, diabetes, heart disease, hypertension, IBDs. In fourth year you will have this inflammatory bowel diseases, intellectual disabilities, mood disorders, obesity, many such are there which are there because of the chromosomal problem. So one portion we have finished, few more we shall be seeing how it is there. There are chromosomal disorders are there and how it can be there. There are various testings are possible. We have seen FEAST and other tests are there. Many other testings are available, and accordingly one can understand. All genetic disorders, they do not directly result into death, but there are no known cures for genetic disorder. On that we shall have one bigger topic, we call it as a gene therapy. The efforts are going on to have a gene therapy, that is whatsoever faulty gene is there, that can be replaced with the correct gene, but still it is not as developed as it was believed. Almost 20-30 years has gone on that, but so far no big what we can say big success has not yet been there. So there are many like that, muscular dystrophy, Huntington's disease, and they do not show any sign till adulthood. All of a sudden it is there. Some during the active time of the genetic disorder, patient mostly rely on the maintaining or slowing the degradation or improving the quality of life, but maintaining the patient autonomy. There are many diseases are there like that, which show. I know one person who was very intelligent, got good marks, went to medical college, went for PG in orthopedics, and all of a sudden he has got a disease where it was all the muscular dystrophy type, all the muscles were going down, and later he become almost no good signs, almost mass of life without any movement and like that. So these are there. When it will come back, nobody knows. As I told you, gene therapy is there, but this is an ongoing battle. I told you, not big success is there, but not treatment are there, and therefore all the maximum treatments, maximum options are there, which are only around the treating the symptoms of the disorder, because gene therapies are not as substantial, as effective as it should have been. Few more things about this we shall be seeing about various gene and their problem. Yes, I'll give you a glimpse. You can go for list of genetic disorders and you will find very big list there. Okay, and even whatsoever so far found, in how many people it is there, very interesting it is there. These are various disorders are there, on what chromosome, at what kind of gene it will be there, what kind of thing it is, whether it is recessive dominant or what kind of it is there, and what is the prevalence of it, in how many people it will be there. So some are very rarest of the rare, that is example 1 in 2 lakh. Okay, that is a ceruplasminomia, that is serenoplasmin, that is cerumen which is there. Okay, that is changing. Uh, then this one is adenylosuccinate lysis deficiency. It is one in around 78 one in 78 lakh people. Okay, or this is around one in sorry, this is 780 lakh people. This is 195 lakh people, a cardiac outer syndrome. So even it is a challenge for doctor to identify the symptoms and to understand the problem. But fortunately, they could have got the data. See, these are the database which are helping, and on that basis, you can have so many, or you can got the list that it is there, and on the basis, doctors can at least diagnose it what it can be there. Because otherwise, in the country like India, people will go to many babas and something like that without understanding actually what can be the problem. So this is the list of various genetic disorders. Just for your, will not definitely, I am not going to ask any question on this, but just for your information that this much why we are studying genetics, just to to give you this glimpse that yes, these are the diseases, and this can result into problem. So other things, what various types of mutation, will have one session, and then we'll finish this particular topic, that why it is there. Okay. So if you have any difficulties, you can put there.